right now, as you listen to this, there is a 30% chance you're carrying a biological weapon just sitting quietly inside your nose.
Yeah, just hanging out,
Yeah,
completely unnoticed.
exactly, just hanging out. But tomorrow, that exact same microscopic organism could be, the terrifying flesh-eating superbug you see dominating the evening news, or the unstoppable force shutting down a hospital ward.
Right. I mean, we like to think of those as two completely different worlds, right? Like a harmless passenger versus a monster lurking in the dark.
But they're the exact same thing. And that's really the core mystery of today's deep dive. We are looking at Staphylococcus aureus.
It's a genuinely unsettling paradox. We're deeply conditioned to view pathogens as these external invaders.
Yeah, like you touch a dirty doorknob, you catch a virus from a guy coughing on the train.
Exactly. The idea that one of humanity's most lethal bacterial threats is a permanent resident on our own bodies, it forces us to completely rethink what it actually means to be infected.
Okay, let's unpack this, because our mission today is to figure out this ultimate biological Jekyll and Hyde scenario. How does a single microscopic organism manage to be a harmless roommate on a Tuesday and a deadly threat by Thursday?
To get to the bottom of that, we have to kind of throw out the whole idea of a simple bad germ.
Right.
If you look at standard clinical microbiology, S. aureus isn't classified the way, say, the rabies virus is.
Because rabies is a strict pathogen, right? If it's inside you, you have a massive problem.
Precisely. You don't just hang out with rabies, but S. aureus is what we call a pathobiont.
A pathobiont, meaning what it rides the line between being pathogenic and being benign?
Yeah, it's a commensal opportunist. It causes absolutely zero disease in the nostrils of roughly a third of the healthy human population.
Wait, so the definition of a pathogen is actually up for debate. Are we defining it by what its DNA says it is, or by whether my skin is broken?
That's the key right there. Modern microbiology recognizes that a pathogen is defined by context. The bacteria doesn't change its identity. Your physical context changes.
Your immune system dips, or you get a cut.
Right. A physical barrier breaks, and suddenly that quiet passenger becomes a lethal threat.
Which means we can't just define a bacteria by a simple list of traits. Though, I mean, if you open up a medical textbook, you get hit with a wall of jargon right away.
Oh, absolutely.
It describes S. aureus as a gram-positive, catalase-positive, coagulase-positive coccus. To anyone who hasn't taken biology in a decade, that's just a foreign language.
Let's break those terms down because they actually perfectly explain why this organism is such an incredible survivor. Let's start with gram-positive.
That's about the physical armor, right?
Yeah, it refers to the structure of its cell wall. Gram-positive bacteria have this incredibly thick, dense armor made of a sugar protein mesh.
Peptidoglycan, I think it's called.
Exactly, peptidoglycan. This armor is what allows it to survive in incredibly harsh, dry environments.
So it doesn't need to be in a warm, moist body to survive.
Not at all. You could leave S. aureus on a dry hospital tray table or a gym towel for weeks, and it doesn't dehydrate. It just waits.
Wow. Okay, so that's the gram-positive part. What about the rest of that textbook string? Catalase and coagulase.
Those are its chemical weapons. So catalase positive means it produces an enzyme that neutralizes hydrogen peroxide.
Wait, like the stuff you pour on a cut?
The exact same stuff. If you've ever poured hydrogen peroxide on a cut and seen it bubble, that's oxygen being released to kill bacteria. S. aureus can literally disarm that attack.
That is wild.
Okay.
And coagulase positive.
That means it can force your own blood plasma to clot around it. It essentially builds itself a little invisibility cloak out of your own blood proteins.
Incredible. And then the last word, coccus, just means it's spherical, right?
Yes, spherical. And it grows in these very distinctive grape-like clusters, which actually brings us to how we discovered it in the first place.
Right, because before modern genetics, bacteria were essentially invisible phantoms. How did we isolate this specific cluster and realize it was the culprit?
We have to step back into the surgical theaters of the 1880s.
The early days of microbiology.
Yeah, a Scottish surgeon named Sir Alexander Ogston was trying to solve a massive problem. This was the era right after Joseph Lister introduced antiseptic techniques to surgery.
Using carbolic acid to clean wounds, right? Listerine is named after him.
Exactly. Lister's methods were revolutionary, but Ogston noticed that even with these new antiseptics, patients were still developing awful pus-filled abscesses.
Wounds were still going bad, and nobody knew why.
Right, so Ogston took the pus from these human abscesses and put it under a microscope. He saw these little spherical bacteria clumped together.
Like the grape clusters.
Exactly. Because of those shapes, he named them Staphylococcus, deriving from the Greek word staphyle, meaning bunch of grapes.
And he proved they caused the infections by injecting them into mice, right? dovetailing with coccus postulates.
He did. He watched the exact same infections develop in the mice.
But the full scientific name we use today is Staphylococcus aureus. Where does the aureus come in?
That came a few years later, in 1884, from a German surgeon named Friedrich Julius Rosenbach. He managed to isolate the organism in a pure culture.
Okay.
And when he grew it on solid agar plates in the lab, he noticed the bacterial colonies had this striking golden yellow pigment. He added the Latin word aureus, which translates to golden.
Golden grape clusters. I mean, it sounds beautiful in a weird microscopic way.
It does have a visual appeal, but what's fascinating here is that golden pigment is far from just cosmetic. The pigment is a molecule called staphyloxanthin, and it functions as a highly effective antioxidant.
Wait, an antioxidant, like the health benefit that people talk about in blueberries or green tea?
The exact same chemical principle.
Are you serious?
Yeah. When your white blood cells corner a bacteria, one of their main tactics is to blast it with a massive burst of reactive oxygen species. It's essentially biological bleach.
And the bacteria uses the golden pigment to protect itself.
Exactly. The pigment acts as a chemical shield, absorbing and neutralizing that oxygen attack. It literally disarms our immune system's heavy artillery.
That is just brilliant evolution.
It really is. Rosenbach also noticed a different non-pigmented cousin of the bacteria on the skin that was much less aggressive.
Which we now call Staphylococcus epidermidis.
Right. So even in the 1880s, scientists were beginning to see the difference between the aggressive golden staph with its antioxidant shield and the quiet background staph.
Okay, so we have this golden armored survivor living in a third of the population's noses. But how does it actually make the leap? If it's sitting quietly, what is the physical mechanism that turns it into an infection?
The defining principle here is colonization leading to autoinfection.
Autoinfection, so you're infecting yourself.
Exactly. Because it lives on us, the vast majority of these dangerous infections aren't caught from someone sneezing on you on the subway. You get a scrape or you undergo surgery, and your own colonizing strain physically relocates from your nasal cavity to your bloodstream.
So location matters more than identity. The zip code changes, and suddenly it's a threat.
That's a perfect way to put it, which makes perfect sense of why hospitals are so obsessed with pre-surgical screening.
Right, if you go in for a routine knee replacement today, the first thing a nurse does is swab your nose.
They're actively hunting for nasal carriers. If you test positive, the hospital puts you through a decolonization protocol.
Like with antibiotic ointments?
Yeah, mupirocin in the nostrils, and you wash your body with chlorhexidine soap. The goal is to temporarily eradicate the staging ground before the surgeon makes a single incision.
But let's follow the bacteria's journey for a second. Say the barrier is breached. The bacteria gets inside the tissue. It can't just float around in the bloodstream aimlessly, right? It would get flushed out or eaten.
Right, it needs to hold on. So it expresses a family of specialized surface proteins that allow it to anchor to host tissues. Biologists gave these proteins a rather clunky acronym. MS-grams.
Right, I read about MS-grams. It sounds like a terrible acronym.
Yeah.
But functionally, is this basically like microscopic Velcro?
Microscopic Velcro is a perfect analogy. These MS-gram proteins are designed to specifically recognize and tightly bind to the molecular building blocks of our own bodies.
Like what kind of building blocks?
Things like collagen in our connective tissues or fibrinogen in our blood. This is how the bacteria physically anchors itself to a heart valve, causing a devastating condition called endocarditis.
Where it literally chews away at the heart tissue.
Yeah, or how it anchors to bone tissue, causing osteomyelitis.
So it velcros itself to your heart valve. Obviously, our immune system is going to notice a foreign invader latching onto a vital organ and sound the alarm, right?
Oh, it sounds the alarm immediately. But S. aureus has evolved two phenomenal evasion tactics once it anchors down. The first is the biofilm.
A biofilm, what does that look like in practice?
Well, once a cluster of these bacteria attach to a surface, and they're especially fond of artificial surfaces like that prosthetic knee joint we mentioned, or an IV catheter, they begin excreting this thick, slimy, sugary matrix all over themselves.
So they build a biological bunker.
Exactly.
They just hunker down in the slime. I imagine that makes it incredibly hard for white blood cells to get to them.
The slime physically repels both immune cells and most standard antibiotics. Often, the only way a doctor can cure a biofilm infection on a medical implant is to physically, surgically remove the infected implant.
Wow. And you said there was a second evasion tactic.
Yeah, and it's even more sophisticated than the biofilm. It relies on a surface molecule called protein A.
Okay, protein A. The immunology behind this is fascinating.
It really is.
Because normally, our immune system pumps out antibodies, which look like little Y-shaped flags, right?
Hmm.
The prongs of the Y grab onto the bacteria, and the stem of the Y sticks out as a signal for our white blood cells to come and eat the invader.
That's the standard immune response, yes. But protein A flips the script. It's designed to bind to the stem of the antibody, the FDC region, instead of the prongs.
Wait, so it grabs our antibodies by the wrong end?
Yes.
It's like a criminal grabbing a security guard's flashlight and pointing it backward into the guard's own eyes so nobody else can see them.
That is exactly what it does. It completely disables our targeting mechanism. The bacteria coats itself in our own antibodies, but because the stems are hidden, the white blood cells just swim right past, completely oblivious to the threat.
It's molecular camouflage.
Perfectly camouflaged.
Hiding in plain sight. But, hiding doesn't explain the massive tissue damage in those flesh-eating headlines we talked about.
Mm-hmm.
A hiding bacteria doesn't destroy a lung or rot away skin. How does it go on the offensive?
Because hiding is just the first step. Once it's secure, it deploys a deeply concerning toxin toolkit.
Okay, a toolkit.
And the important nuance here is that not every strain of S. aureus carries every weapon. Different strains acquire different genetic instructions, like downloading different apps for different jobs.
What kind of apps are we talking about here?
One of the most notorious is called PVL, or Panton-Valentine leukocidin. This is a pore-forming toxin.
What does that mean, pore-forming?
When the bacteria releases PVL, the toxin seeks out our white blood cells and physically inserts itself into their outer membranes, punching a hole straight through them.
It literally punches holes in our immune cells.
Yes. The structural integrity of the white blood cell fails, its internal contents leak out, and the cell dies. The bacteria destroys the very soldiers sent to kill it.
That is brutal.
It is. Strains carrying the PVL toxin are strongly linked to severe necrotic skin infections, that flesh-eating presentation, and a rare but devastating condition called necrotizing pneumonia.
Which often strikes young, otherwise completely healthy people, right?
Yeah.
Because it's turning their own strong immune response into a liability.
Exactly. And that manipulation of the immune system brings us to another major weapon in the toolkit: superantigens.
Superantigens.
Specifically, toxic shock syndrome, toxin 1, or TSST1.
Okay, toxic shock syndrome. This takes us back to the massive outbreak in the 1980s linked to Rely superabsorbent tampons, right?
Yes, unfortunately.
That caused absolute panic because healthy young women were suddenly dying.
The synthetic materials in those specific tampons created a highly absorbent, oxygen-rich environment. It was the perfect incubator for S. aureus to rapidly multiply and pump out TSST1.
But the deeply terrifying part about a superantigen is that the bacteria never actually has to enter the bloodstream to kill the host, does it?
No, it doesn't.
How does it cause a massive system-wide shock if it's localized in one spot?
The bacteria stays put, but the toxin it produces absorbs into the bloodstream. And superantigens break the fundamental rules of our immune system.
How so?
Normally, our immune system is highly specific. A regular antigen will only activate about 1 in every 10,000 T cells. It's a targeted response. But a superantigen acts like a biological skeleton key.
A skeleton key.
Yeah, it artificially binds to receptors on T cells, indiscriminately activating up to 1 in 5 T cells in your entire body all at once.
Wow. So instead of a targeted strike, it sets off every alarm bell in the body simultaneously.
It triggers what we call a cytokine storm. Your immune system effectively panics, overreacts, and floods the body with inflammatory signals.
Which causes the blood pressure to drop.
Exactly. Massive inflammation causes blood vessels to leak, leading to crashing blood pressure, a distinct rash, fever, and rapid multi-organ failure. The body destroys itself in response to a toxin pumped out by bacteria that are just sitting locally in one contained location.
It's just terrifying. But here's where it gets really interesting. We've been talking about how this bacteria uses toxins to attack our immune system when it gets inside the body. But some of its toxins don't even need the bacteria to be alive to hurt us.
That's right.
Let's talk about the infamous buffet potato salad and staph food poisoning. I always thought food poisoning meant I accidentally ate a bunch of bacteria, they set up camp in my intestines, and my body had to fight off a gut infection.
That is how a salmonella or an E. coli infection works. But staph food poisoning is fundamentally different. It is not an infection at all.
It's an intoxication, right?
Exactly. It's an intoxication, meaning the illness comes entirely from a preformed chemical.
So how does that happen at a picnic?
Well, the bacteria produces a specific type of weapon called an enterotoxin. And these enterotoxins are remarkably heat stable.
Heat stable.
Yeah. Imagine someone who's a nasal carrier of S. aureus wipes their nose, then mixes a batch of potato salad with their bare hands. They leave that salad out on the buffet table at a summer picnic for a few hours.
So bacteria are multiplying in the warm mayonnaise.
Right, and they're pumping out large amounts of enterotoxin.
Then someone realizes it's been sitting out in the sun, gets worried, and decides to, I don't know, aggressively microwave it or cook it back down, assuming the heat will sterilize it.
And cooking it thoroughly will absolutely kill the bacteria. Every single S. aureus cell will die.
But the toxin survives.
Exactly. That heat-stable enterotoxin remains completely intact. It doesn't break down. So you eat the cooked potato salad.
Even though there's no live bacteria in it.
Right. Because you're ingesting a preformed toxin, you don't have to wait days for bacteria to colonize your gut. The toxin hits your system immediately, triggering violent vomiting and severe diarrhea within 1 to 6 hours.
That is incredibly fast.
It is one of the fastest onset foodborne illnesses on the planet, orchestrated by a bacteria that is already dead.
The ultimate phantom strike. So, you have an organism with microscopic Velcro to anchor itself, slime bunkers to hide in, backward-facing camouflage to trick antibodies, toxins that punch holes in our white blood cells, superantigens that cause our immune system to self-destruct, and heat-proof toxins that ruin our picnics.
It's quite the resume.
Naturally, humanity decided to drop the atomic bomb on it, antibiotics. But this triggers arguably the most consequential debate in microbiology, right?
Yep.
An evolutionary arms race that we seem to be actively losing.
It is a relentlessly fast evolutionary race. If we connect this to the bigger picture, we have to look at the timeline. In the early 1940s, penicillin was mass-produced.
It was a miracle drug.
Absolute miracle. Patients with systemic staph infections, who previously had a near 100% mortality rate, were suddenly walking out of hospitals fully cured.
But the bacteria didn't just take the hit.
It adapted almost instantly. We have to remember how bacterial evolution works. Bacteria replicate every 20 to 30 minutes.
So fast.
Right. When you introduce an antibiotic, it's like a meteor hitting the dinosaurs. You wipe out the millions of vulnerable bacteria. But if just one single mutant happens to have a genetic quirk that lets it survive, it now has zero competition and unlimited resources.
It just multiplies exponentially.
Exactly. By the late 1940s, less than a decade after penicillin was introduced, hospitals were already finding staph strains that had evolved to produce an enzyme called penicillinase.
Penicillinase, what does that do?
This enzyme acted like chemical scissors, literally snipping the penicillin molecule in half before it could harm the bacteria.
So the miracle drug was rendered useless in a matter of years.
Yes. So the scientists had to go back to the drawing board to find something the scissors couldn't cut.
And they did, right.
They did. In 1959, they introduced methicillin, a modified antibiotic designed specifically to be immune to that destroying enzyme.
Okay, problem solved.
Not quite. The incredible selective pressure of the drug forced the bacteria to outsmart us again. By the early 1960s, just a couple of years later, resistance to methicillin emerged in the UK.
Right.
Yeah, this gave birth to a term that everyone has seen in the headlines. MRSA, methicillin-resistant Staphylococcus aureus.
Wait, people always say superbugs are a modern problem caused by doctors overprescribing pills today. Are you saying this bacteria was rapidly mutating and outsmarting our absolute best medical technology back when my grandparents were kids?
Exactly. I mean, while modern overprescribing definitely accelerates the spread of these resistant strains in the community, like the MRSA outbreaks we see spreading through shared gym equipment or locker room towels, the underlying capacity for resistance evolves on the exact same timescale that we introduce the drugs.
Because the selective pressure forces rapid mutation. I want to clarify something about MRSA, though, because I think the media has created a massive misconception here. When people hear the acronym MRSA, they tend to think it's an entirely different alien species of bacteria that is infinitely more aggressive than regular staph.
That is perhaps the most dangerous misconception in public health. MRSA is not a different species.
Right.
It is normal Staphylococcus aureus that just happens to have acquired one specific gene, known as the MECA gene.
Okay, what does that one MECA gene actually do?
Well, normally, antibiotics like penicillin and methicillin work by binding to proteins on the bacteria's surface, jamming the machinery it uses to build its cell wall, which causes it to burst.
Okay, so the drug targets a specific protein.
Right. The MECA gene provides the instructions for a mutant version of that protein. The new protein works perfectly to build the cell wall, but its physical shape is slightly altered, so the antibiotic simply cannot latch onto it.
Oh, wow. It's like changing the locks on the doors.
Changing the locks is exactly what it is.
So MRSA just means it changed the locks to block methicillin. What do we call the normal kind of staph?
We call it MSSA, methicillin susceptible S. aureus. And here is the crucial point that gets lost. MSSA can be just as invasive, just as tissue-destroying, and just as deadly as MRSA.
Really?
Yes. Resistance to drugs and virulence, how sick the bacteria actually makes you, are independent traits. They do not automatically travel together.
Okay, that makes sense.
An MSSA strain equipped with that pore-punching PVL toxin can be much more dangerous to a patient than a MRSA strain that lacks a robust toxin toolkit. The only difference is which pills the doctor is able to pull off the pharmacy shelf to try and save you.
That is incredibly sobering. And the arms race didn't stop at methicillin either. When MRSA became widespread, doctors had to turn to heavier artillery.
They turned to vancomycin, which for a long time was considered the reliable last-line defense for serious MRSA infections.
Right, the drug of last resort.
But in recent years, we've seen the emergence of Visa and VRSA strains with intermediate or complete resistance to vancomycin.
And the mechanism of how S. aureus achieved this resistance is truly alarming, isn't it?
It's terrifying. In a documented case inside the body of a single patient, S. aureus literally stole the vancomycin resistance genes from an entirely different species of bacteria called Enterococcus.
Wait, how does a bacteria steal genes from a completely different species?
Through a process called horizontal gene transfer, bacteria have these small circular pieces of DNA called plasmids.
Plasmids.
Think of them like microscopic USB flash drives that carry a few specific files, like the blueprints for drug resistance.
And they can just share them.
Yeah, bacteria can physically connect to each other and pass these USB drives back and forth, even across species lines. The S. aureus simply plugged into its neighbor, downloaded the blueprints for vancomycin resistance, and became a nearly untreatable superbug.
It crowdsourced its own invincibility, which completely explains why there is such a massive debate in global healthcare right now. If it's this adaptable, do you try to wipe it out entirely? I mean, some experts argue hospitals should universally decolonize everyone who walks through the door, just in case.
While others say that costs a fortune and we should only target high-risk patients.
It's a terrifying balancing act. When you look at the whole picture, it is wild to comprehend. To quickly recap our journey here, location, not identity, is what makes it dangerous. MRSA is a trait, not a species. Disease can be pure toxin like food poisoning in hours or toxic shock. Biofilms and protein A allow it to outsmart both our devices and our immune systems, and the evolutionary resistance race is just relentless and fast.
It's the ultimate lesson in biological context and adaptability. And understanding the mechanisms of how S. aureus operates fundamentally changes how you navigate the physical world.
Yeah, it really does.
It explains why not sharing a towel at the gym is a real medical necessity, not just a preference. It explains why you should always avoid the lukewarm potato salad at a picnic.
Definitely taking that one to heart.
Right. And it demystifies the intense protocols behind a routine surgery. Or why a doctor treats a simple red angry boil on your arm with such serious caution.
It really makes you look at your own skin and the microscopic world living on it a little differently. Thank you so much for guiding us through the biology of this.
My pleasure.
But before we wrap up, I want to leave you, the listener, with one final thought to mull over. We just talked about how S. aureus acquired its vancomycin resistance by physically plugging in and stealing genetic USB drives from a totally different organism, Enterococcus.
Right.
But earlier, we mentioned its quiet cousin, Staphylococcus epidermidis, the non-pigmented background bacteria that lives all over our skin, which just happens to be an absolute master at forming those impenetrable, slimy biofilms on medical catheters. If S. aureus is already actively in the business of stealing genetic secrets from its neighbors, what happens if it fully downloads the ultimate biofilm blueprints from its quiet cousin? Could the ultimate, completely untreatable superbug actually be born from a microscopic genetic handshake taking place right now, quietly, on the surface of your own skin? Something to think about next time you get a paper cut. Thanks for taking this journey with us. Keep questioning the invisible world around you, and we'll catch you on the next deep dive.